we have Dr. Stephen Gray coming up next, who's a professor at UT Southwestern Medical Center, and he's going to talk about steps to initiating a SURF1 gene therapy clinical trial. He'll discuss the latest developments in this effort and give an overview of the steps to take this sort of lead candidate through IND enabling studies. So how do you go from this type of lab data to a new drug and a first in human clinical trial? I'll hand it over to you, Dr. Gray.
All right, thank you. Glad to be here. Thanks for inviting me to speak today and share our work with gene therapy around SURF1 related Leigh syndrome. And I want to first acknowledge that this work was really spearheaded by Qingyuan Ling, the last speaker, while she was a postdoctoral fellow in my research lab. And we've been doing our best to kind of continue the work on this as she's moved on to her exciting new paths at UMass. Let's see. So these are my disclosures. And so first of all, probably don't need to introduce adeno-associated viral vectors very much, but just to highlight that this is a pretty ubiquitous vector that's used now for a number of disease applications, hundreds of clinical trials, and several FDA-approved treatments. And it's essentially a very staple capsid protein shell that's used as a carrier and for a genetic cargo. And and really in terms of delivering a gene to the central nervous system, there's not any well-established vector that's been sort of used for in vivo gene delivery besides AAV to broadly target the whole brain. So this is currently sort of the gold standard for doing gene transfer to the central nervous system. Okay, so of different causes of Leigh syndrome, SURF1 is the most common. It's a ubiquitously expressed intermitochondrial membrane protein, but it is expressed out of the nuclear genome. And its role is basically to facilitate heme A insertion into complex 4. It's required for complex 4 assembly. And when you have an absence of SURF1, then you end up not assembling functional COX-4 very well. So you have reduced COX-4 activity, which leads to decreased ATP production, increased lactate, reduced endurance capacity, and energetic demands. So patients are estimated to have about 20% of COX activity remaining. So our work on this started back in 2018 and resulted in a nice publication that we were excited about in 2021 that Dr. Leigh was the first author on, where we developed an AAV9 vector carrying the SURF1 gene to treat SURF1 knockout mice. And so this study showed favorable safety in mice, and it showed efficacy in terms of increasing COX activity in the mice and decreasing exercise-induced lactic acidosis. So we were excited about this. We engaged with a company partner that was interested in this at the time to move this towards a clinical trial. But unfortunately, as we got to the stage of doing formal GLP toxicology studies with this vector in rats, we encountered some unanticipated toxicities. And again, none of these were seen in our mouse studies, but when we moved on to rats, then we saw a degree of dorsal root ganglia toxicity, these sensory neurons that run down beside the spinal column. But I think more concerning was we saw a significant amount of heart pathology in terms of actual degeneration of cells in the heart, infiltration of mononuclear cells. And I think the heart toxicity in particular was was something that gave us a lot of pause and made us think that this was not an appropriate treatment to try to take into patients despite the unmet need in the patient community. This original vector design was a self-complementary AAV genome. It used the chicken beta-actin hybrid promoter, which is a relatively strong promoter. And so basically at this crossroads where we decided we did not want to do anything to take this vector forward, we had to go back and redesign the vector. And the solution here was to engineer different versions of this vector that would incorporate moderate or weak expressing promoters. And these are based on this promoter called JET, which is a small synthetic and relatively weaker promoter. And so you have a base JET promoter and then a slightly stronger version that incorporates a synthetic intron. And I will say that this JET promoter is something that we've advanced into clinical trials for diseases like giant axonal neuropathy starting 10 years ago, and CLN7 Batten disease started 4 years ago. And then this version of JET with the intron is something that we've taken into a clinical trial for spastic paraplegia type 50. So there is a translational path and translational experience taking these general designs into humans. And I should say, if we treat human fibroblasts with an AAV2 vector carrying these different constructs, then on a Western blot, we actually can't detect any protein expression with the JET or the JET-I promoters. So the expression is quite weak. But if we look at a functional improvement in the fibroblasts in terms of looking at increasing COX activity, then we see that these weaker promoters are able to rescue COX activity to the same extent as our original stronger version. And we would expect about 50% of the cells to receive the transgene in this assay, and that corresponds nicely to about a 50% restoration of COX activity in the whole cell population. So we went on quickly to move this construct in vivo, and I'm showing data from the sort of the middle, the moderate expressing construct with the JET-I promoter. And first of all, if we looked at the expression, and this is delivered into SURF1 knockout mice by a lumbar intrathecal delivery route. This is the delivery route that we would be planning to use in humans. And these were all mice that were injected at 10 days old. And so comparing the two constructs, like our original with the CBH promoter and the revised one with this JET-I promoter, we saw significantly reduced expression in the heart, and then sort of modestly reduced expression in the brain, including the striatum, or the whole brain of the striatum. We think that we're seeing increased expression in the liver, probably due to a degree of liver toxicity that maybe we weren't recognizing, or unoptimal expression. So we did see, if we looked at increases in COX activity in the cerebrum, the brain as a whole, then with the JET-I promoter, the CBH promoter, we saw basically about the same degree of improvement in COX activity. And then the cerebellum, we saw a trend of increase that wasn't quite significant. But then we saw increases in the heart and skeletal muscle. And I should say in the heart, even though the expression with the weaker promoter, even though the expression was weaker, we saw better restoration of COX activity. Again, I think in keeping with this notion that we were just expressing too much with the CBH promoter, that was actually kind of overshot and was detrimental to improving the COX activity. We didn't see as much benefit in the liver, but that may be an effect of us dosing mice at 10 days old, after which they undergo like a lot of growth in the liver and kind of a dilution of the vector genomes. But at this point, we had kind of engagement here in the brain, the heart, and skeletal muscle, all critical targets for Leigh syndrome. The other aspect of efficacy that we were looking at that we had investigated before with the original construct was where you run the mice to exhaustion on a treadmill, and then you can measure exercise-induced lactic acidosis. And this is measuring the difference. So their baseline blood lactate levels are basically the same between wild type and knockout mice, but then you see this elevation in lactic acid in the knockout mice after exhaustive exercise. So there wasn't any difference in the overall treadmill running time, although there was a slight trend of decreased running time with the knockout mice, and there was a trend of restoration upon treatment. But when we're measuring the blood lactic acidosis, then we see that this is elevated in the knockout mice and that it's normalized upon treatment. Okay, so that's from an efficacy perspective, and I should say, from a safety perspective, everything still looked fine in the mice, but we did take on a study evaluating this revised sort of weaker version of the SURF1 vector, doing lumbar intrathecal injections in normal rats in my lab. This is we did a high dose only, and this dose in rats corresponds to roughly a maximum feasible dose if you were going to extrapolate this to humans. And this was a dose at which we saw toxicity with our original vector design. But in this case, we dosed mice at 8 weeks old, did a necropsy at 28 days old, and we basically saw no toxicity. So no changes in weight or body condition. A comprehensive histopathology analysis didn't show any of the heart toxicity or dorsal root ganglia toxicity that we saw with the original vector design. So, so overall, again, this initial vector design that we published with the CBH promoter, it was effective in the SURF1 knockout mice, but it showed unacceptable toxicity in normal rats. By revising the vector design to use this JET-I promoter instead of CBH, then we were able to achieve the same degrees of efficacy as with the original construct. In some cases, like the heart, we saw better efficacy. And very importantly, our revised SURF1 vector design with this JET-I promoter was well tolerated in normal rats. So we feel like now we've settled on a design that that is both safe and effective, that we could take forward to a human trial. So we re-engaged with the FDA. We had a prior pre-IND meeting with them around the original construct before we ran into all these toxicology findings. And so with this revised vector design, we held a new pre-IND meeting February of earlier this year, to go over this new vector design and a path forward. And the takeaways are basically that the FDA said yes to everything that we wanted to do, and we were broadly in agreement with them about a path to take this into patients. They agreed that the treatment held that we provided preclinical evidence of a direct prospect for direct benefit to pediatric subjects. They agreed with us that we could conduct a pivotal toxicology study in rats. So there's no large animals or non-human primates needed. They agreed with our plan on manufacturing and releasing the clinical drug, and we got agreement with them on a basic design of a clinical trial. And importantly, in the meantime, as we've been working on this therapy, my lab had some separate investigations looking at developing improved sort of next generation versions of AAV9. We have one that shows some improved potency over AAV9. and that we thought would be advantageous to use here. And we proposed that to the FDA, and they were actually in agreement with us that we could switch to this improved version of AAV9 with a set of minimal bridging studies that we proposed and that we have ongoing right now. So overall, our next steps, this is sort of a schematic or diagram of the steps to try to get to a first-in-human clinical trial. We've conducted our proof of concept studies demonstrating the efficacy and the safety of this. We held a pre-IND meeting with the FDA and got guidance and agreement with them on what these next steps should be to submitting an investigational new drug application. And so right now we're at a step of trying to identify a drug manufacturer, conduct the toxicology studies, and manufacture the drug and prepare the IND. These are steps that will take at least a year, and right now we're still trying to identify funding sources to carry out this work. So with that, I want to acknowledge my team, in particular, it's Ching Lan, who you should recognize from the earlier talk, and also Matt here in the back, that contributed very heavily to this. And definitely appreciate all of our funding sources and the engagement from the family foundations, especially Casey Wolben, the Cure Mito Foundation. Happy to wrap up and take talks or take questions.